structure becomes unmistakable. All these cellular changes happening. Lots of dynamic cytology going on here. So these are the seminiferous tubules. Two most complete ones right here, and then parts of ones over here. All we'll do, start at the wall, say that begins the spermatogenic process. Nice big round nuclei, very active mitotically. So these are the spermatogonia, sometimes just called gonial cells. They divide, and then these develop to start showing their chromosomes. You see that worm-like appearance in the nucleus. Those are spermatocytes. I only care that you identify them as spermatocytes. Don't worry about them being primary or secondary. But what you're doing here genetically is preparing them for meiosis. Reductional division so that this and this becomes this. Much smaller nucleus. These are the spermatids. The tids. Now beginnings of the gamete cells, the cells that can achieve fertilization once they've matured to look like this. Boat-shaped nucleus tells you it's the spermatozoa, and that these are now flagellated. They actually don't move until they're at the end of the vase. Just flow right along with the seminal fluid. So that's the spermatogenic line. We start with spermatogonia, spermatocytes, spermatids, spermatozoa. And then know that this is all nurtured by a cell that encases these cells. The Sertoli cells, or nurse cells. How do you tell them? The nucleus. There's the nucleus of a Sertoli cell. Oval, a little higher would be clefted, and even the nucleolus inside. These are Sertolis. And that you get a good sense of how they're supposed to work with this diagram where the cells are embedded in the cell membrane of those cells. That's why you can't easily pick them out. Here's the clefted nucleus and that nucleolus. And that you need to know the special function where these cells are all holding hands all the way around the lumen of the Sertoli cells. Anything in the blood is screened off or away by these barriers called the BTB. So all I need is those three letters, BTB. Ensures that the male's immune system doesn't destroy their end cells. Otherwise, the species would be in big trouble. Recall or keep in mind the BTB for Sertoli cell functions. And then you finish it with cells that are in between the seminiferous tubules. One wall here, another wall of one over here, couple, 3 cells in between. Those are the lighting cells, or interstitial cells. Nice bunch right there, lighting cells. One over here, there's a lighting cell right there. You should know what they're responsible for. Testosterone producers. Primary production of the male sex hormone. All right. Everything you need for testis histology. And then just how important is the spermatic cord running through the inguinal canal. Any kind of compression or pressure on this tells us the testis now can be lost. That's where all the blood vessels come from. Everything to and from the testis is through the spermatic cord. So with the complaint of increasing testicular pain, not something to be taken lightly, a matter of 4 or 5 hours, and all is lost. Tissue goes necrotic. Gangrenous has to be removed. I say the male definitely knows there's something wrong. It doesn't sneak up on you in terms of this greatly increasing pain as the tissue is now being destroyed. And then the histology within the spermatic cord. Pick up on the muscular tube that's in the cord. So there's the vas deferens, big, thick muscular wall. Let's say it's peristalsis that moves things along in here. That little tiny lumen. This is a nerve trunk, so all the nerves to and from. And then these are all the blood vessels. All these holes are a vascular plexus. I only want you to name it all together as a functional unit. So all of these blood vessels call the tympaniform plexus. Why is it structured like this? To radiate heat from the arterial blood. Knowing that spermatogenesis goes unstable above 94 degrees Fahrenheit, it's important to cool that 98.6 blood coming from the abdominal cavity before it hits the seminiferous tubules. So one of these would be an artery, and then all the others are testicular veins with cool blood coming back, intended to cool the arterial blood. So think of the vasculature of the spermatic cord as this functional plexus and tympaniform. All right. Accessory glands for the male, prostatic tissue. This is seminal vesicle and no ID. And this is 50, 55-year-old male showing pretty good precipitates in the little pockets. So I say think of glandular sponge, a lot of it being fibromuscular bands, and then corpora amylacia could be mistaken for colloid. But when you look around and you see all of this muscular banding, can't be thyroid. So that's got to be the inert glycoprotein in the prostate. Corpora amylacia or just CAs. No pathology whatsoever here, that's natural. And then a better stained specimen, really giving you a sense of these fibromuscular bands, smooth muscle fibers here. Little alveoli with this age-related deposition, glycoprotein. Even a little one starting there, but that's a really good example and can be initially confused with colloid. Seeing all of this should keep you away from thyroid. Make sure you get a really good look at that banding. Thyroid, and the figure 8 for tunica albuginea. Great, thick, strong connective tissue within. So this keys you in on the penile cross section. That's the dominant structure. Encased within is the soft, delicate, erectile tissue is the cavernosus, even showing its arteries. And then knowing this structure, jagged opening, the urethra must be kept open with its own protective collar. Sort of a counterweight to the cavernosus, where the spongiosum ensures that the urethra stays open or patent at all times. It's the figure 8 that tips you off. And then the cavernosus, spongiosum, urethra. All you need for that histological section. All right, slides you haven't seen before, especially relative to these. Or could you tell if this is male or female? So here's the urinary bladder. Is this the front or the back? Look at where these are going. Yeah, they always go into the back side of the bladder. So this is a front view, anterior view. So now you've got the right ureter, left ureter coming down underneath to the back. You then have coming out the base, right here, urethra. Yep. So here's the cutoff end of the urethra, which is encased in this. Glandular mass, prostate. There. So this is a cut open prostatic area where the urethra is seen to be right down through the center of it. This is why any enlargement of the prostate immediately starts blocking the urethra. Got to get in and cut it out. So that tells you it's got to be a male prostatic tissue. Peeking out from behind. Could you tell what those are? Little ears. Yeah, seminal vesicles. Yeah. I did bend them down a bit. I would always show you them from a posterior view. You can get them to peek out a little bit if you push it forward. Seminal vesicles, right and left. But this was used to show just how tight the prostatic tissue is to the urethra. And any enlargement now, you get the spinning blades like a rotor rooter to now cut out that impinging tissue. Gives the male the willies, you think about it. Pass that spinning blade up through the urethra to get to that impinging tissue. Quite a bloody procedure, even for a few days afterwards. All right, never saw this before. Can you tell where it's from? Let's say for next week, histology, you're thinking renal, endocrine, or male repro. We have those three histological areas to organize. You look at this and you say, is that renal, endocrine, or something to do with male repro? What do you think? It's renal cortex, sure. This could look like islets, but you got all these tubes. As soon as you see all those tubes, now it's got to be renal cortex. These are your renal corpuscles. I would never have you identify anything more than that. Magnify it a lot more for other structures. All right.